Hey everyone, welcome back to Policy Punchline. Here at the show, we interview scholars, policymakers, and business executives about some of the most urgent issues and frontier ideas in our world today. I'm Princeton senior Tiger Gao. Today, I'm very excited to introduce Professor Robert Langer, who is an acclaimed chemical engineer, professor, and investor in biomedical technology. He is one of the 10 institute professors at MIT, which is the highest honor that can be awarded to a faculty member. He has written more than 1,500 articles with 1,400 patents issued and pending worldwide, and have been licensed to over 400 companies. He is the most cited engineer in history with an H index of 283 and over 300,000 citations around, according to Google Scholar. So his inventions are estimated to have affected over 2 billion lives. And his most recent public work involves the coronavirus vaccine created by Moderna, which is the biotech company he co-founded. So as you can see, Professor Langer is probably the biggest deal that we've had so far on Policy Punchline. So Professor Langer, thank you so much for joining us today.
My pleasure. Thank you for having me.
And as our audience may know, I am more of an econ generalist guy. I don't know anything about science. So I've brought on two of my most brilliant friends, Arjun and Michael Senka. Both of them are also aspiring intellectuals who are pursuing PhDs in computer science. They've applied to MIT, but they haven't heard back. So we don't know what's going to happen on that front. But Arjun, Michael, welcome to the show.
Thank you.
Thanks for having me.
Professor Langer, perhaps we can start with a very broad question, which is, could you tell us a little bit about your early journey? How did you become interested in science and engineering, and chemical engineering in particular? What drove you to pursue a PhD in the field when you were an undergrad at our age?
Yeah, well, I have to admit, I didn't really think it through that carefully. You know, when I was a little boy, they had these what are called Gilbert chemistry sets and Gilbert microscope sets, and they were fun to play with. And so that got me somewhat excited about science and even chemistry. But, you know, when I was in high school, I was good in math and science. So my parents, or my father and my guidance counselor, told me I should become an engineer, even though I didn't really know what that was. So, but that's what I did. I applied to engineering schools, and I went to Cornell. And my first year, the only course I really liked and did well in was chemistry. So that made me decide to be a chemical engineer. Like I said, it wasn't that deep a thought. In terms of my next steps, then I, you know, when I was finishing, I mean, different classmates of mine did different things, but the only jobs I really got were like running a chemical plant. And I thought I would be terrible at that. It'd probably blow up the place. So, you know, being a graduate student struck me as, you know, a way to, you know, really get maybe more in-depth understanding of some additional options that I might do. So I ended up doing what your colleagues are talking about, is going to graduate school.
Makes sense. And so then, you know, after your graduate studies, you talked about how, you know, I think that the petrochemical industry was recruiting heavily, and you know, so you were offered many traditional chemical engineering jobs after your PhD, and instead of taking those offers, I believe you decided to find a position in medical research working with Judah Folkman. So I think we're excited to get into sort of the research that you did there, but first, what motivated you to make that decision and to choose medical research over kind of the more traditional offers that you had?
Yeah, so what happened when I was a graduate student, a couple things. One is, you're right, like a lot of my friends, everybody was going into oil companies. There was like this gas shortage then, they had a lot of openings, and I got like 20 offers, but I wasn't excited about it. I mean, you know, you go there and then you think about what you'll be doing for the rest of your life. And, you know, they'd be talking to me, boy, if you could just increase the yield of this one chemical by 0.1%, that would be great, you know, be worth tons of money. But, you know, as I started thinking about that, I just didn't want to do that. I didn't know what I wanted to do exactly, but I knew it wasn't that. I wanted to be able to have some impact, I felt, on the world, whatever that might be. And when I was a graduate student, when I was doing my thesis, one of the things I spent a lot of time doing was helping some other people start this high school for disadvantaged or working class high school students. And I really loved that. I got very involved in creating new chemistry curricula and new math curricula. So actually, I started thinking that I might want to do something like that, but maybe in a broader way. So what happened is I remember seeing an ad one day for City College of New York to create new chemistry curricula. And I thought that would be really good. And I wrote them, but they didn't write me back. But I liked the idea. I started applying to all these other schools to do the same thing. A lot of them weren't even very good, but I wasn't in the right box, like the chemistry education box, because there's a major you can do in that, and that's not what I did. I was a chemical engineer, so I none of them really got back to me. So that was kind of discouraging, but it did make me think that what I wanted to do was use my chemical engineering education to help people. So that made me think about medicine, and I started applying to a lot of medical schools and hospitals, and they didn't get back to me either. But I like that idea. And then one day, one of the people in the laboratory I was in, Barry Bunow, he suggested to me that there was this surgeon named Judah Folkman, and that he sometimes hired sort of unusual people. So I wrote him, and he was nice enough to get back to me, and that's how I ended up there.
Wonderful. Yeah, so I think it'd be lovely to sort of jump into the research that you were doing with Judah Folkman. And so our understanding of it is that you were trying to come up with some inhibitor that can stop blood vessels from growing in order to, or rather even come up with a factor that can control blood vessel growth with applications to reducing the growth of tumors. And so so the goal was to try to find inhibitors that stop the tumors from essentially vascularizing and growing bigger. And so how did this idea first come to light? And I think our understanding was that while most people at the time didn't actually think it was possible, so I'd love to hear more also about why you decided to stick to the idea and why you thought it was possible.
Sure. Well, first, I mean, that idea of angiogenesis, that and blood vessel growth being important to tumors and other diseases, that was Dr. Folkman's idea. I thought it was a very appealing idea, partly because I didn't know much about biology. It was widely criticized at the time, and I didn't know that either. But I felt like basically the idea for cancer is that normally blood vessels, well, first, if you take a tumor and it grows abnormally, it might grow as a three-dimensional mass. But if it didn't get blood vessels, it could never really get larger than a millimeter cube, because you can't get nutrients to the center and you can't get rid of waste. But his theory was that the tumor made some substance, which he called tumor angiogenesis factor, and it diffused to the surrounding blood vessels, which were normally quiescent, but then in the presence of these molecules, that all of a sudden the blood vessels would start growing and growing, and they'd grow right to the tumor and they'd feed it. So they'd solve the nutrition problem for the tumor. And that enabled it to get bigger, bigger, and bigger. And also, it could spread through those blood vessels later. That's metastasis, which often kills people. And so that was his idea. He hired me to, I guess, prove his idea, and in so doing, isolate the first angiogenesis inhibitor. And like I say, I thought this was, to me, being a chemical engineer, certainly made a lot of sense. Now, maybe if I'd known more biology, I would have thought, like other people, that maybe it didn't. But and like I say, he was widely criticized, I mean, at that time, and actually for many years. But it turned out he was right. And so in terms of me trying to take that idea and make it happen, there were a couple of things that I did. One was I had to find something that could stop blood vessels from growing. And there we looked at cartilage, which doesn't have blood vessels. And I'd start getting tons of cartilage from slaughterhouses and scraping the meat off of them and extracting different things. But then the other real key issue was creating a bioassay, for it. That was really there really wasn't a good way to study whether you were stopping blood vessels, because there's almost always background blood vessels, and it takes a while for them to grow. So here we created what we call a rabbit eye assay, and other people use mouse eyes or rat eyes. But basically, it was always along the same idea, that if you put certain tumors in the eye, they would cause over a several month period blood vessels to grow from the edge of the cornea to the tumor. So you could actually quantitate that with an ophthalmic microscope. But the key to creating the assay was that the molecules we were isolating were pretty large molecules. And nonetheless, if you put them in the eye right away or used eye drops, they diffuse away right away. So what was critical was creating something that was biocompatible that you could put in the eye and that could deliver these large molecules. So that was the other big thing. So I worked on sort of two projects that were, somehow connected. One was isolating substances that could stop blood vessels from growing, and two, creating this bioassay, which involved creating the first drug delivery systems for macromolecules.
So can we just quickly define, can we just quickly define the word bioassay, and so that we understand like what these two problems were and how each one was working together?
Sure. So bioassay is a way to study some factor. And in this case, the bioassay was a way to study blood vessel growth. So what we wanted to do was have a bioassay that would enable us to look at how fast the blood vessels were growing, and would, if we isolated something that could stop blood vessels, would we really be able to see it, and would we be able to quantitate it, and maybe someday even purify it. So that's the bioassay. And then, like I say, we were studying, our initial thought was cartilage as a source of this inhibitor.
So Professor Langer, as you explained, you really struggled a lot for the project on blood vessel growth because you first had to develop this bioassay to study blood vessel growth, and there wasn't one available. And it took you months to develop the polymer systems and bioassay, and you had to separate many different molecules, and almost all of them didn't work out until you finally successfully isolated the substances that could control the blood vessel growth. And you've talked about this story many times, but I would love to, I guess, go back to that time and picture myself or let our audience picture themselves in your shoes back then, because so many people doubted you and it was a long struggle. Why did you stuck with the idea? I mean, I guess that the bigger question here is, what is the process of drug discovery, both for you today and when you were much younger? Because as a young scientist, a lot of times the interesting part of your early career is that you didn't know too much about biology, about chemistry. So you said a lot of times you felt like you were in the dark.
Yeah. Well, first I thought this was just a really important problem. I mean, I really felt and it turned out that was right, not that I realized it fully at the time, but I just felt like, boy, if I could do this, if we could do this, stop blood vessels from growing, I mean, that would be just this huge deal, because it could open up new possibilities for cancer therapies and maybe other therapies too. So I just kept working on it for several years. I mean, I'm pretty stubborn, and Dr. Folkman was a great role model. He was just a very encouraging person. And it meant something to me to do it for him, to do it for the lab. So there were really two parts to it. One was doing it, and even though it took several years to do it, that was one part. But I think the second part was, even after I did it, it was really, people were very skeptical, both on the angiogenesis part and on the drug delivery part. The drug delivery part even more, because all the much better or more well-known scientists than I, chemists and engineers, said that what I was doing was impossible. Like, we published, like I say, in this journal Nature in 1976. Actually, that's probably, I didn't mention that. So we did publish in 1976 the assay for the angiogenesis inhibitor and the isolation of these first inhibitors in the journal Science in 1976. We published in 1976 also the idea that about these materials that could deliver macromolecules, and that we published in Nature. But both of those, there was a lot of skepticism for many, many years. Like, for example, the idea that materials could deliver large molecules, people didn't think made any sense. Just like they'd say, like, how could any of us walk through a wall? We're just too big to get through the wall. And also, we use certain kinds of solvents, like organic solvents, and people said, well, that didn't make any sense either, because the large molecules that I was putting into these materials were like peptides, proteins, and nucleic acids. And this may come up later, because nucleic acids are DNA and RNA. And it was the first time that anybody had ever put them in tiny particles and then delivered them. Of course, and that's the basis of a lot of these vaccines that you mentioned in the beginning. but still people were super skeptical of me doing that in in the paper, going back, close to almost 50 years.
So as you mentioned, once the inhibitor of blood vessel growth was discovered, there is the challenge of delivering it since it is naturally broken down by the body. Can you explain in more detail the drug delivery system you invented to deliver the inhibitor to the tumor in a controlled release fashion?
Yeah, so the specific thing we did is I looked at a lot of different materials. I ended up using certain polymers, plastics, and most of the designs I used didn't work. Most of the materials didn't work either. But I did, so I kept experimenting. It was more like Edisonian, like, just trying different materials, trying different designs, concentrations of the drug, coating the materials with other materials, and so forth. Most everything didn't work. Either the drug didn't come out or it came out right away. But eventually I did find certain materials. In this case, it was what's called an ethylene vinyl acetate copolymer that worked pretty well, and that's how I got started. Later on, I'd find others too, and other people, lots of people over the years have, taken this a lot further than where I was when I started it.
So yeah, and so on that point, your lab continues to work on drug delivery. So I'm curious, how have the drug delivery systems developed in your lab evolved since the first version? Have the basic principles stayed the same? Have more recent designs become more complex? For example, I think recently your lab released a Starshaped drug for release in the stomach. So we'd love to hear about how the systems have evolved since the first iteration.
Yeah, well, it's evolved a lot, not just because of me, but I mean, now there's giant industries doing this stuff. I mean, one way to think about it is in 1974, when I started, I don't think there were any of these advanced drug delivery systems. Now, these delivery systems are used almost by everybody in one way or another. I mean, there's microspheres that you inject to treat cancer, to treat mental health diseases, like you hear about opioid addiction. I mean, those are some of the major ways to treat that. I mean, the COVID vaccine, every COVID vaccine based on messenger RNA or even a protein uses nanoparticles. If you have heart disease, you get what's called drug-eluting stents. So, I mean, almost everybody in the world gets these things. And then we've been also doing a tremendous amount of work with the Gates Foundation to try to come up with medicines for the developing world too. That's at an earlier stage, but that's also very important. That's a big part of what our lab works on. So that's one way of looking at it. Another way of looking at it is just the technologies. The early work we did were model systems, with model drugs. But what would happen over the years is, real drugs would get put in for injectability. But over the years, we and others would work on transdermal systems that you put like patches on your skin, systems that you might put in the eye to deliver eye medication, new inhalers to deliver drugs to the lung, other types of things that you deliver to the nose. We're working actually on ways of delivering things to the ear for people who are deaf or things like that, can't hear well. It goes on. And then there's even more advanced things like better vaccine delivery systems. You mentioned the Starshaped system, which I could show you even if it's helpful. But it's but basically we have ways of now, which are in clinical trials, swallowing pills that might last for, rather than a day, which has been the maximum so far, maybe even a week or a month, and then we even have things that we're designing that are pills you could swallow that might be able to deliver insulin and things like that. And it goes on and on. I mean, and it's hardly just us. I mean, I've had probably 4,000 students and postdocs go through the lab, many of whom are professors all over the world. There's tons and tons of companies started by many, many people. And, they've been key to all of this.
Well, I think we'd love to see the Starshaped drug.
Sure. I'll show you a couple of things. The Starshaped one, this is an example. So I can just show you what we do. is this was done by Gio Traverso, Alex Abramson, and others. But the idea is that you have the centerpiece, which is made out of a degradable polymer, and then you have these spokes. And each spoke could contain a drug or a different drug. We've actually put 3 HIV drugs in, each of different spokes. But basically the idea is with this type of chemistry, you can just fold this up. And this is an early one, but you can fold this up like this. You can put it, and when you fold it up, you can put it in a capsule like this. And this just fits in the capsule. So the idea is you swallow the capsule gets in the stomach, which is acid. And because it's acid, it will now dissolve this capsule. As soon as it dissolves this capsule, this will now pop out, just the way it was before. But the beauty of this design is that if you look at your anatomy, so here first you go to your mouth, then your throat, then your stomach. But before you get from your stomach to the rest of your intestine, there's something called the pylorus. The pylorus is like a hole, and it's not even that big. So this shape will sit on top of it. And the beauty of this particular shape is it's so big, it can't pass through, but it's so open that food and other things can pass through. So it doesn't cause blockage. And then at whatever time you designate it, you can, this centerpiece will dissolve. And when it does, everything will pass out. But what that enables you to do is deliver, you can take one pill and it might last for the entire course of treatment, or maybe you take a pill once a week or once a month. So we're working on all these things. We're working with the Gates Foundation on a once every 2-week pill that could wipe out malaria in certain communities. And then we started this company, Lindra, which is with Gio Traverso and Amy Schulman and Andrew Bellinger, and I think that now we must be in like 7 or 8 different clinical trials for different diseases.
I'm particularly fascinated by this issue of control, right? So I guess the problem was that these molecules would dissolve on their own. So you encapsulate them, they release in a controlled fashion. It seems to me that I guess the ideal level of control would be if you have the molecule and then it's sort of being navigated through the body, and as soon as it detects a tumor, then it sort of releases the appropriate amount of the inhibitor that's necessary to deal with that tumor and then moves on, right? That's kind of the ideal level of control. So how far are we, so what are the current methods of control that are used in these modern drug delivery systems for releasing the correct amounts at the correct locations, and how far are we from that ideal case?
Yeah, well, I don't think we're at that ideal case, yes, but there's sort of 2 different aspects of control. One is or maybe even 3. One is timing, second is kinetics, and a third is location. So each of those are different levels. I would say from the standpoint of timing, there's different ways, when the release would start, and even developing ways to start it. I think we've, you're reasonably far along. In terms of kinetics, getting almost any kinetic pattern you want, I'd say we're pretty far along. In terms of location, that's more complicated. I think if you do what's called local delivery, there are different ways that we and others have done it. Like one way is we have these implants that neurosurgeons put in the brain, so you get high concentrations in the brain for the tumor and low concentrations in the rest of the body. Another example, which I mentioned before, are drug-eluting stents, which is probably the major way to treat heart disease today. And there you take pretty toxic drugs that are normally used in chemotherapy, like Taxol, and put it on this stent, which is like a Chinese finger puzzle, and then it locally delivers the drug right to the blood vessels, and it keeps them open. So I think for situations like that, we're also pretty far along. But the idea of what I'll call a magic bullet, which is kind of closer to what you said, you inject something and all of a sudden it finds its way to exactly where you want it to go and now releases its drug. I think that we're still, that's still elusive. I mean, there are situations where you can take advantage of drugs having leaky vessels. The body having leaky blood vessels near a tumor. So it may preferentially accumulate in a tumor. there are examples of certain targeting approaches that you can use for different parts of the body, like the liver, so that you could direct a nanoparticle to the liver. But really broadly being able to target, I think still we have a long ways to go.
Do you think that some level of wireless technology could help? Maybe this is very out there, but if you were able to almost remote control the particle inside the body out, with kind of wireless communication, is that something that's even feasible, or I'm just curious?
I think parts of it are feasible. In fact, one of the things that we've done is we've created these like implantable microchips that you can do. We actually even published this in Nature, and we even brought it into clinical trials. But those are macroscopic, and so you could. So those are macroscopic, but you can do exactly what you said wirelessly. You can cause certain wells to open and release the drug at different times. So that's an example of controlling, of wirelessly dealing with one of the three situations that I mentioned in terms of the timing. You can, for example, have a cover on the wells of the different chips, and you can wirelessly open those chips if you want. And when you do, drug can come out. So we actually did a clinical trial for patients with that had osteoporosis and could deliver parathyroid hormone really on demand. But the idea of now having a super tiny particle that you could control wirelessly, I mean, I certainly don't rule that out. But the question might be, it has to be so tiny, and then you might need to image it, and you might have to figure out the navigation. And I'm not sure even about the power requirements, for something so incredibly small. But it's certainly something that we've thought about. But the first one that I mentioned, which is a little bit easier problem, is still pretty hard. So we're still working on that more.
So there's one paper in particular of yours that I saw recently that I thought was a very interesting application for drug delivery research. This was, if I understand it correctly, you and your lab were developing a drug delivery system to address chemotherapy for PDA, a pancreatic cancer. And this was really cool to me just to see how purely research in drug delivery was used to help chemotherapy and otherwise very painful treatment process. So I'm curious to hear from you for cancer treatment as a whole. How much closer can we get to what one would call a cure for cancer purely through drug delivery research?
I think you can help cancer through drug delivery research, but a cure is tough. I mean, I don't think, several different systems that we've developed and different people have adapted are now widely used for different types of treating cancer, but I don't know that they're cures. Cures are really a hard thing. So I think drug delivery can help an awful lot of cancer treatments, but I think curing it is going to be is very, very hard. And I think it's going to be ultimately a combination of better understanding the biology and coupling that with things like that you just mentioned. But even there, it may be that someday, and this would even be an improvement over where we are today, that cancer will be more like heart disease, that you'll manage it, that you won't necessarily cure it, but you'll keep it under control in more and more situations. We'll have to see. I mean, certainly significant advances in cancer have been made, but I still feel there's a long ways to go in terms of cures.
Professor Ling, I think Michael was just trying to figure out when we can expect it, so he can jump in investing in some companies beforehand. The purpose of Policy Punchline is mainly to get some insider information about what's on the frontier.
You could just invest in all the companies that have come out of our lab.
Exactly. Well, we will go there in a bit, but perhaps another big area aside from drug delivery system is tissue engineering. That is another field that you have devoted a lot of time on. You and your colleagues developed a 3D synthetic polymer scaffolding, which to an economics major like me already sounds fascinating. That could, do that to the new skin, bone, blood, organs can grow. And could you help us define tissue engineering a little bit, explain how it works, why it works, and what are some of the specific problems that tissue engineering research could address?
Sure. Well, so it must have been, close to 40 years ago, Jay Vacanti, who's a friend of mine, and he was head of the liver transplant program at Children's Hospital, was talking to me about how we could help babies that he was treating who were dying of liver failure. And so we came up with this idea of creating scaffolds, taking maybe the patient's own cells or other cells. And the idea is if you injected those cells at random, nothing happened. But when we put them on the scaffold, again, with the right scaffold, the right media, they were able to be smart enough to reorganize themselves and create a new tissue or organ. So that was, like I said, the early 80s. But over time, that's developed into many different things. And now you can make artificial skin for burn victims or patients with diabetic skin ulcers that way. And there are many, many other tissues and organs in clinical trials, a number of companies doing that. And there's all kinds of variations that that people have developed. And then people are using it not only for, making new tissues and organs for patients, but even organs and tissues on a chip, which might improve drug testing and, reduce testing in animals and people.
So to understand the principle behind it, let's say we took capillaries, which develop tubular structures. And so what you're saying happens is that if you just put capillary cells together, they don't naturally form the tubular structures, that there has to be some sort of scaffolding that exists. And so if that's true, how do you discover that scaffolding?
Yeah, well, actually, what you just said is right. In fact, one of the people in Folkman's lab, Dr. Folkman's lab, they actually in vitro took capillary cells and put them on like a Petri dish with certain media, and it actually formed a capillary tube. That was part of the basis for Jay and I thinking that maybe we could do this, that some of these things happen naturally. So part of it is just finding the right substrate. There's different materials. Part of it is sometimes intelligently making that substrate, because there may be literature information saying that certain amino acids or certain structures may improve the ability of certain cells to adhere to a material. Part of it is figuring out the right media. Part of it is figuring out the right way to grow the cells. I'll give you an example of that, which Laura Nicholson, who is one of our fellows, she wanted to make new blood vessels. And people had tried to do that before. This is small diameter blood vessels. And so the idea that she had was that, not only did we need the right material, and Jinming Gao was another one of our postdocs, did a lot of work on that, but we even needed the right reactor conditions. So generally, I don't know if any of you have watched people growing cells, but usually they put them on Petri dishes or other things. They kind of just sit there. Sometimes people have had some motion, but none of that really worked. So Laura's idea, she said, people have tried to make blood vessels, but blood vessels don't just sit there like one of these Petri dishes. Blood vessels in your body are hooked up to a pulsatile pump like your heart. So she did that. She created a pulsatile pump and so that went like at 170 beats per minute. And we published in Science journal that you could make new blood vessels that way, and it worked really well. And then she actually started a company on that. I'm on the board of directors. That might be one to look at. I think they're doing like a SPAC, they're doing they're in phase 3 trials or with some pretty good data to make new blood vessels. And and they published an article in Lancet a couple of years ago showing some of the data. So so everything matters. It's the cells, it's the tissue, it's the bioreactor conditions, all these kinds of things.
Amazing. I did actually do some wet lab research myself in high school, but I personally wasn't very good at it. I think one time I turned on sort of the gas instead of the vacuum or something, and I think I ruined a whole week's worth of experiments for the whole lab. So I realized that my future wasn't there personally. But yeah.
It sounds like all you guys are doing okay.
Yeah. So yeah, just to kind of dive deeper, what are the current obstacles and limitations in doing this tissue engineering? So how does the approach compare in success to, for example, transplanting an organ from a fully formed organ from another human?
Yeah, well, that's a good question. First on the organs. So the biggest issue on transplanting the organs is that, we just don't have enough, right? There's giant organ shortage on everything you do, liver, everything. And then in many cases, the organs, haven't really worked well when you have transplanted them, say like in a pancreas or something like that. So, so I think this is at an earlier stage, obviously, but it's like I say, already led to skin. In the case of blood vessels, like I say, she's already in advanced clinical trials. Other companies that some spun out of our lab are in pretty advanced clinical trials for spinal cord repair, for hearing loss. I hope that there's now a lot of activity for creating new pancreases, one of which came from our lab. So I think that there are many more possibilities of how you could use tissue engineering to create these new tissues and organs. You could use the model that I said with real cells and scaffolds. Some places, including what Laura's done, is to, and Humacyte is to decellularize them after you've used it to serve its purpose. Other cases, people are, as you may know, are looking at stem cells. We've done some of that. Other cases, and again, a lot of this was done by Jeff Karp and myself. Jeff was one of my postdocs, now professor at Harvard, is sometimes you can even isolate specific factors that can cause certain types of stem cells to grow. So you can give those as a drug. One of the companies we started on that, which is doing quite well, is Frequency Therapeutics, which is trying to treat hearing loss. And it can, and these factors or substances can cause the hair cells in the ear to grow back and multiply.
It's really amazing. It's really amazing to hear all those extensions, but do you see this research having any impact beyond the medical community? So for instance, there's a growing demand for lab-grown meat. Arjun is a vegetarian, and he has been trying to indoctrinate Michael and I, eating more beyond burgers, whatever, for a long time. So,
oh no, you're absolutely. People are doing that. Yeah. In fact, it's funny. A number of famous people. This must have been 15 years ago. Like would ask me if you could ever do that. But over the years, like Shulamit Levenberg, she's one of my former postdocs. She's in Israel. She started a company to use tissue engineering to make meat. And there are quite a few others too that are doing it. I mean, I must be 5 or 10 companies that are trying to make meat that way. And then to your point, there's a company, Modern Meadow. My son actually worked there one summer. They're making leather, from it. And then, like I say, there's all these organs and tissues on a chip. Quite a few companies doing that. Tara Biosystems, which is one that I'm a director of, and was started out of the University of Toronto and Columbia by Gordana Vanyak and Melissa Ratzek, who were postdocs and students of mine. And they're, they have like a heart on a chip, so they're doing drug testing, for all these different companies. So there's all kinds of things that are getting done. And there's other companies doing, Vivitex is doing a gastrointestinal tract on a chip. Actually, SBS in Texas is also doing heart toxicity on chips. A number of companies have started livers on a chip by Linda Griffith and Sangeeta Bhatia. There's a lot of different ones.
So to pivot to another popular topic, there was a paper from your lab that incorporated machine learning and deep learning in particular recently for optimal drug injection. There seems to be a trend in some scientific computing communities to introduce neural network in their models, particularly when numerical simulation of high dimensional things is important. So I'm curious to hear your thoughts on what kind of role artificial intelligence may have in your field. Do you think neural networks have a big future in drug delivery research, for example?
I do. I think actually, the example you gave is a good example, but one of the things I'm particularly excited about that I've talked with some of the people at MIT about are could you apply that to chemical structures and really learn about it that way? So let me give an example. We've done a lot of work on with Dan Anderson, who was one of my postdocs, now professor at MIT, on synthesis, creating what I'll call large libraries of materials for different things. So one example is it's making lipid nanoparticles, and it turns out that the type of which, by the way, are used in COVID vaccines. They're used in for delivering what is called siRNA, and that's already led to treatments. But let's say you make 1,000 nanoparticles, maybe 10,000 nanoparticles with 10,000 different lipids. So it turns out that there at least, maybe there's 3 or 4 problems that you might want to address. One is, how do you make the lipid nanoparticles more and more potent? Say you're trying to deliver siRNA. So we work with Alnylam, a local company that I was on the scientific advisory board of, and between our students and the people at their company, which also included some of my former students, we were able to, over like a 10-year period, increase the potency of these systems by a factor of 10 to the 4th, 10,000. So one question is why? What structures did we pick that enabled that to happen? So that's one problem. Another problem, which you actually alluded to before, is some may target, if you wanted to target to certain cells in the body, again, some of these nanoparticles work better than others. A practical example that we could give, which we haven't done, but it's in the news all the time, is the Moderna vaccine you can store at room temperature for a month, but the Pfizer one you have to put under really very, very low temperature conditions. So anyhow, the point is that you could have libraries of all these things, and we do have libraries of many of them. But then could you use machine learning to say, well, okay, here's certain lipids that work really well, here's ones that work poorly, here's one that worked okay. Could you use machine learning to figure those problems out? Could you use them to actually predict the next generation and the next generation of say lipids or polymers or whatever that could help you on potency, that could help you on targeting, that could help you on storage and so forth? And I don't limit it to just that. Like we've developed with Dan, polymers that are materials that are more and more biocompatible in the body. We've published a lot of this in different nature journals, and it led to another company, Sigilon, which is also doing well. And we've come up with sort of what I'll call super biocompatible materials. We get almost no fibrous encapsulation, which, by the way, is going to be important for breast implants, lots of other things. So could you, again, when you have these libraries of materials, take machine learning and plug in, okay, here's structures that work great, here's ones that don't work at all, and again, learn how to make even the very best chemical structures. So those are some things that I've been real interested in. We've started talking to people who are, very knowledgeable at MIT, like Regina Barzilay, on these kind of machine learning approaches and AI things. So I think there are a lot of exciting things that can be done. The big issue is you need really good data sets to do these. If you had contamination or impurities, that might make it much harder to do it. But I think there's a lot of really good potential in these areas.
By the way, Professor Regina Barzilay will come on our show in March, so it would be great to hear her thoughts on these issues as well. But
she's doing great work. That's good.
Yeah, Professor Langer, I noticed that as we spent the past 40 minutes or so going over your research, you mentioned a lot of your colleagues, postdocs in your lab, and your former students. So I think perhaps it would be nice for us to learn a little bit more about your lab, because the Langer lab is MIT is the largest biomedical engineering lab in the world, maintaining over $10 million in annual grants and over 100 researchers, and so many wonderful companies have come out of this hub of research. And business school professors have always said that if there were more Langer-like labs that focused on high impact research, the United States would realize its enormous potential for creating wealth and doing good for society. So perhaps we can talk a little bit more about your lab?
Sure, of course. Basically our lab, it's very, I just has a couple features that I could highlight. One is it's very interdisciplinary. In other words, like my background's chemical engineering, but we have people in the lab probably with about 10 different disciplines, from, graduate students and postdocs and MDs in different areas. So, and it's been like that for a while. So one thing that maybe distinguishes is its interdisciplinarity. We also have probably the largest undergraduate lab of any place at MIT too. You know, we have had lots of great undergraduates working in the lab doing what are called UROPs. That's a MIT kind of term, undergraduate research opportunity program. But I think it's the interdisciplinarity and also it's the idea of doing basic curiosity-driven research in science and engineering, but not just leaving it at that. In other words, do make those discoveries, some of which we've talked about, but it's also wanting to see those discoveries ultimately get used by patients and people. And, you know, and I guess I found that if you, in many cases, if you're not your own champion, you know, nobody else will be. So, you know, so that's where we've started companies and we've pushed things along to the point where, you know, I like to think that people feel, you know, it's whether it's us or somebody else, it will make an impact in people's lives.
You were previously joking that we should just invest in all the companies that came out of the Langer lab, which to me is a really unique feature because you've previously spoken about the 3 Ps of launching a company from university research, which stands for platform, patent, and paper. So how do those words define your approach? And would love to hear a little bit more about that front.
Yeah, well, what we do in the lab, again, my goal in the lab is a couple-fold, just to do really good research and to train the very, very best people. But I also mentioned that it's been important to me and I think many of the students to see if they do this work and it's working somewhat well, that it gets used. So I guess the first thing is, in my experience, if you make a discovery or you make an invention, it's probably going to have more impact if it can be applied to many things, right? Like when I showed you that STAR system before, you could apply that to virtually any drug. And if you took messenger RNA, you could apply that to many different problems. So that's a platform. And I think a platform can have a great impact. In fact, if the technology is good, even if one of the things that you try to make happen doesn't work well, or several of them don't work well, others may work well. And so, but it's just the potential to have an impact with drug delivery or tissue engineering or you know, nanoparticles. I think it's just greater if you have much greater if you have a platform. The chance of making an impact is much higher. We always publish everything we do. I mean, it's that's important to me for the students. It's part of MIT policy. In fact, I would say that over the last 5 years, we've probably published over, on the average, of about 1.3, 1.4 papers a month in Science and Nature journals. So we not only try to publish, we try to publish in high impact, difficult to publish places, which I found has been very helpful to getting the students get good jobs. And, you know, and so that's then, you know, because universities like to see that. And as you pointed out, we started companies, and that gets to the 3rd P, which is the patents, that unfortunately in medicine, everything you do in medicine costs an enormous amount of money. You know, Tufts, I think, has an estimate that it costs close to $3 billion to make a new drug with all the testing that has to be done. And investors will not put in money unless they feel that there's some protection. And that's where the patents come in. So we try to do that as well. So those 3 things always come up.
One thing that you mentioned was how your lab is extremely interdisciplinary, and that's something that sets your lab apart. It seems like that was a struggle earlier, and that you had difficulty personally being a chemical engineer and doing biomedical research. And at least from my perception at Princeton, it seems that a lot of university faculty and administrators now strive to have more interdisciplinary faculty, projects, and researchers. Do you think that's unique to a few schools, or do you think in general academia has become perhaps more open to interdisciplinary research that draws in multiple fields to solve important problems?
Yeah, I think it's been an evolving thing. You know, so when I went to Children's Hospital in 1974, I was the only engineer in the entire hospital. It certainly wasn't common. And in fact, if you looked at universities then, And probably even now, but I think it's a little different. I'm not sure exactly when I visited Princeton last, but it's at most schools, there's probably, I'll make this up, but let's say there's a building for chemical engineering, there's a building for economics, there's a building for English, but there's not a building for, that is that interdisciplinary. In other words, the buildings are largely single discipline. So I think that it has changed, I think, but I still think it's not that common. And I don't think it's that simple. I mean, I personally love interdisciplinary work, but I don't know that there's just one right way to do it. It's just that's what we do. But I feel, and I think being interdisciplinary for me, being a chemical engineering, wanting to see the things we do, make an impact the way I feel they've done, and I hope continue to do, I think that's been important because the problems we're trying to address and the discoveries we're trying to make, they don't just come from one thing. So I do feel it's certainly changed from 1974, but I still think the rule is more single disciplinary than multidisciplinary. But Phil Sharp and myself and Susan Hockfield, who was president of MIT, and Tyler Jacks, we've all talked about this concept of what we'll call convergence. And I do feel there's more and more of that. And I think there's been more and more interest in that. I know Phil and I wrote an article in Science, must have been in 2011, on that very issue. And I feel, more places are thinking about doing that. And I think that's a good thing. But like I say, I don't know that there's just one right way.
So on the topic of there being no one right way, I'm interested in hearing more about the fact that this dichotomy between academia and the private sector. So, in particular, when we were talking about your research, you mentioned that a lot of your research has been spun off into companies that you're leading or play important roles in. And so you're renowned actually for spinning your research into private sector enterprises. And so I'm curious to hear, what do you think are the upsides and downsides of academia in the private sector, and why is it important that scientists engage in both?
Well, again, I think it's most important that scientists do what they love and that they feel is good for them and for their students. Again, I don't know that there's just one right way. But the way I personally look at it is I love doing the work in my MIT lab. I love working with the students. That's always been my first priority and still is. But myself and actually a lot of the students who've worked on this, you do work on something for 5 or 10 years. You want to see it have the biggest impact on the world. And so you can only go so far in a university, right? You could publish the paper, but you can't do clinical trials, or it's not very easy to do clinical trials, I should say. It's very hard to do manufacturing and so forth. So, so the companies, I think, have done a great job at doing those kinds of things, and others to take an idea that might be a discovery or an invention and now bring it to the world. So I personally have felt it's a good thing, and a lot of my students have really wanted to do that in postdoc. So I feel like both are important.
But by the way, Professor Lang, I just want to say on the topic of interdisciplinary topic, I've been talking to the econ department about teaching classes in the art museum for a long time. They just wouldn't do it. They said it's too distracting. So
no, that's a good example. There's a huge resistance to, and there always has been a huge resistance to doing what you just said and what I did. In fact, we didn't really get into it, but when I was applying for faculty positions in 1977, no chemical engineering department in the world would hire me because they felt the ideas I had, didn't make any sense because they were more in the bio area.
So I'd love to hear a little bit more about this dichotomy between private sector and academia, because a lot of times the role of investors is often seen as taboo in academic circles. So even in something like economics, if you talk about going to finance, the practitioners are often seen as very distant from the academic people, whereas you guys, you really try to work hard to making your research and your students' research accessible to the public, and helping fund biotech companies over the years. And this intersection between engineering and finance is really expanding over the past few years as more scientists begin to enter this investing space. So do you believe that the two disciplines will eventually continue to become more closely intertwined in the future? Are there any ethical considerations or concerns you have on your mind?
Yeah. So so I don't know that we invest. I think we invent, and then what the sort of the model, at least that we've used, and I think that lots of people now use, is that we make a discovery or an invention, and then we feel like, well, maybe this could be a company, and then there are what are called venture capitalists, right? And there's a number of venture capital firms, and in fact, number of my students work in venture capital firms too now. But so the venture capital firm wants to make an investment in our invention and start a company on it, and so that's how it gets started. And some of the early employees of the company are the students who made those discovery and inventions in our lab, and then it grows from there. So I think that. But investors play a huge role, because if without them we wouldn't have the company, especially in the medical area that takes so much money. And so I think that they play a huge role, and usually they're on the boards of directors. Sometimes some of the scientists are too. I've been on a number of boards of directors. But I do feel like it's kind of almost a marriage in a way, right? The investors are putting in the money. They're sometimes helping on the management, and they have to keep raising or putting in more money and providing guidance. And then the scientists and engineers and the clinicians, they're moving the technology forward and forward. And in fact, my feeling when I look at any of the companies, startup or otherwise, usually you'll have some business people like the CEO. You'll have some medical people like the chief medical officer. You'll have some finance people like the chief financial officer, and maybe some business people who do deals of the small company with the larger companies. And then you'll have lots and lots of scientists who are really making the discoveries, doing the work. You might have a group of manufacturing people as well, depending on the size of the company. So it's really a marriage of many different skills.
So at what point are you able to say that research is ready to be spun off into a company? Yeah.
Great question. And what I've tried to say, I'll give you two answers to that. One is, what I've tried to do is say, well, okay, if we've met the following criteria, we've got a platform technology, we've got a paper in a good journal, we've got a patent, that's a very good patent, ideally what I'll call a blocking patent, but at least it gives you freedom to operate. Again, if it's in the medical area, that we've got in vivo data, not necessarily humans, but in vivo data. And finally, that there's some people in the lab that really want to do it, that they feel committed to really wanting to start this company and work there. And one of the main reasons I've done it a lot of times is that that's what my students have wanted to do. That's their dream. And so that's one way of thinking about it. Another way of thinking about it is, I think that what goes on in the MIT lab is largely research, real research. And when you start the company, I think then you're at least doing some significant development. In other words, when you hear the phrase R&D, that's research and development. And so I think that it's not to say that companies don't do research, they do, but they're going to be doing development because ultimately they have to turn out products.
So
both those ways.
You've previously talked about the importance of funding and fueling innovation. Is it how much of money is the issue in creating these breakthroughs for certain diseases? Does spending more money directly correlate to a higher likelihood of success? I suppose that it's conceivable that if a country pours $1 trillion into a Manhattan Project scale endeavor, like curing cancer, breakthroughs, could they inevitably happen? Or is biomedical research a field that money ultimately plays a relatively secondary role?
Well, I think it plays a very important role. I think the fact that, even like, say, for example, in medical research, the NIH budget over the years has, increased a lot. And I think that that's what the NIH has funded has really led to, I think many of the major discoveries that have come about. I think without having that funding and without having that freedom, I don't think we'd be where we are today in terms of any of the biotech companies. So I think the funding has been critical, and I think it will continue to be critical. That doesn't mean you want to just dump money out. I think you want to use the money judiciously. But I think that even though the NIH is hardly perfect, I think they've done overall very good job over the years of doing funding. And I think other funding always helps. I think what the Gates Foundation has been doing which is more directed, but I think it's been, also quite successful. And I think sometimes companies do funding of academic work, and I think, again, depending on the example, that's been successful. I feel, having been the beneficiary of all three of those, that having had that funding's made a huge difference. And it's not only made a huge difference on the research we do, but it's also made a huge difference in the training, that the students who have worked in the lab have gotten great jobs, and that's, whether it be in academia or in industry. And so I feel all over the world. So I feel the money is really important. I think when I think if a country doesn't put money into basic research, I think that it's going to be very hard for them to do the kinds of things that have happened in the United States.
I'd love to hear more about the work with the Gates Foundation. And I think in particular, what are the challenges that are unique to delivering medicine in I guess, quote unquote, developing countries, and what are the kind of things that you face maybe in those kinds of problems that you might not face in other drugs that you develop?
Yeah, well, I mean, one of the big problems is what's called compliance. In other words, people don't come back for a second injection. They may not take a second pill, but they're different things. Like somebody might not take a second pill because they're just not used to doing that. In the case of vaccines, they may not come back for a second injection just because of the kind of medical care they have in the first place. There's also nutritional problems. There's huge vitamin and mineral deficiencies in the developing world that are certainly less common here. So those are some of the things that have come up, and those are all things that we've been working on different solutions for. And the STAR even was an example of that, because there you could take possibly a pill that could last a week or a month, maybe even a year, and for the entire course of treatment. So, but there are many other examples too. We've been working on vaccines where you can just give a single injection rather than have to come back for multiple boosts. We've been working on new nutritional ways of giving, fortifying different things like vitamin A and iron and so forth. So there's a lot of different problems that come up. And there's different diseases, of course, that people get in the developing world that they don't, like TB and malaria and other diseases. And then sometimes there's other problems just because they don't have good medical care, like babies have acute respiratory distress syndrome. If they were, so we're trying to make simple inhalation therapies for them because they don't have the same hospital facilities that we have in the U.S.
Professor Langer, I think maybe this is a good time for us to be a little bit more forward-looking, because it seems that we are in the midst of a scientific revolution. We've never seen as sort of robust of biotech innovations and capital activities as these days. I think 2019 was a record year. 2020 was also a big record year. COVID obviously made a lot of people realize the importance of healthcare, and a lot of people on Wall Street have been saying that biotech would continue to outperform and would be the next big thing, especially the convergence of DNA sequencing, AI, gene therapy, especially CRISPR gene editing technology that would allow us to cure so many more diseases, and also surfacing mutations in genomic profiles so that we can detect and prevent a lot of the diseases in the long run. So what biotech trends do you foresee coming? What are some of the things on your mind these days?
Well, I think there's all kinds of biotech trends happening and coming. I'll sort of make two types of comments. One, I mean, what you can already see are big advances in areas like proteomics and other omics like lipidomics, glycomics. These are different types of molecules. I also think you see big advances in material science and nanotechnology as they can be used in medicine. As you pointed out, there's whether it's CRISPR or gene editing or messenger RNA or but different genetic therapies I think are very important. I do feel AI could be very important, as you've mentioned, and digital medicine also. There's been a lot of interesting work on, I think cellular therapies are going to be very, very important, whether they're for ways of attacking cancer or regenerative medicine, which we've mentioned before, like tissue engineering. So I think there's all kinds of things like that, and others that I'm sure I didn't mention, things like drug delivery and so forth, but lots of those. But I sometimes think that some of the things that'll be the most important don't have a name yet because they really haven't happened. Like if you ask somebody 10 years ago what CRISPR was, nobody would know. Now everybody knows, and I think that that's a good thing, that people are going to keep discovering things by doing basic research. And some of those basic research things will change the world.
So to talk more about the vaccine now, you were, of course, one of the founders of Moderna, the company behind one of the first approved coronavirus vaccines. A Business Insider headline claimed that Moderna had designed the vaccine in just 2 days. For context, for the viewers, the vaccine development process typically takes at least a full year. In this case, both Moderna and Pfizer sped up the process by relying on mRNA vaccines, which essentially deliver instructions to the body on what type of antibiotics it should make. Could you begin maybe by breaking down the science behind the COVID vaccine and Moderna's approach to this science?
Yeah, so it's actually antibodies rather than antibiotics. But the idea is that so basically there's a central dogma, right? The DNA makes RNA, makes protein. So classically, what people did for vaccines were more like on the protein part, right? And so whether they'd either use inactivated viruses or things like that, or they'd grow up eggs that could make a protein or and so forth. So what Moderna did is start with the messenger RNA. When you do the virus work or the eggs or whatever, it may take a very, very long time to grow up enough to make the vaccine. The beauty of messenger RNA is you could make the messenger RNA and you can do it quickly, but then you give it to the body, you inject it, and the body is actually your factory. The body, since like I said, DNA makes RNA, makes protein, well, you give the RNA and it's going to make the protein. So that actually is a paradigm shift in the sense that now you could make the vaccine extremely quickly and start testing it in people, in very short time, maybe a month. That's what Moderna has been able to do. That's been one of the key pillars of the technology. But there are others too, and some of it gets to what we talked about before, which is the delivery. Because if you just gave messenger RNA to a patient, it would get destroyed immediately. So that's where the delivery again comes in. So what's done is it's encapsulated in nanoparticles, in this case lipids, but it could be others. People are looking at others. But basically it's encapsulated in nanoparticles. You shoot the nanoparticle with the messenger RNA into the patient. It protects it so it doesn't get destroyed immediately. And sometimes you have to decorate the nanoparticles with different molecules like polyethylene glycol, so it will last longer. But the point is you inject it and it goes like into the muscle or wherever. And then now you have the RNA comes out and it starts making, the body starts making the protein, and then the body will start making antibodies to that protein. And if it makes enough, then you'll be fully immunized. Usually you get 2 shots because the first one starts the antibody production, but you start having these, get some memory. And so when you give the second shot, you make even more antibodies and hopefully have high antibody titers. Sometimes you may need a booster shot too, because over time it may wane. So you may need a booster shot depending on the situation.
So what was the reason for the record speed? Was it sort of an investment into a development of a platform that could lead to very rapid testing of vaccine candidates? Like, well, how come it was so fast? It seems it's, especially compared to previous vaccine candidates.
Yeah, well, first messenger RNA is going to be much faster than previous vaccine candidates just because of the nature that I mentioned, that you're using the body as a factory, and that you could design from, literally, if you know the sequence, you can design the mRNA right away, and then you just make it, put it in the nanoparticles, and you're ready to test it. So that was one reason. The other reason really was, I think, Operation Warp Speed. That the government put in a lot of money and accelerated testing. In other words, they were able to do, normally you do phase one, what is called phase one, which is looking at safety, phase two, looking at efficacy. These are both on small numbers of patients, and then phase three, a large number of patients. Here, they move those at much faster speeds than normal. So because of both the funding and the way the regulatory agencies analyze things, they were able to move much, much quicker because, it was an emergency situation. So that's why BioNTech, who work with Pfizer, and Moderna, which I'm on the board of and co-founded, were able to get approval, in well less than a year. But it's a combination of both the science and, I think, this incredible emergency, and then, both the FDA and the government, putting in this funding.
Professor Lang, I just want to quickly clarify this with you. It seems that this is a whole new endeavor because Moderna previously had 0 approved product in mRNA technology, but very soon there should be a few dozens of drugs and treatments in the pipeline, if I recall correctly.
Well, yes and no. So Moderna, even when the COVID virus, even when we started that work, that was actually vaccine number 9 that went into the clinic. There's many others that Moderna has worked on that are in clinical trials. So and so they've probably, so the tests that we did on those clinical trials, they weren't approved. Like I say, you have to go through phase 1, phase 2, phase 3. So, but there were, I think, 8 others at that time that were either in phase 1 or phase 2. So it wasn't like there was no science or no human clinical trials. But in a way, the COVID vaccine leapfrogged over all those others just because of the speed that I mentioned, because of the funding that the government put into it and the movement that the FDA did. Whereas the others went through more what I'll call standard, even though I think it's faster than what had ever been done before, it's still, not as fast as the COVID vaccine because of how, everybody, because of all that increased funding and because of the trials, the FDA.
Right. So it seems like you have these 3 pillars. You have academia, you have the private sector, you have the government, and all 3 seem to sort of converge perfectly and work really well together to solve this immediate crisis. Do you think that the way that these 3 different pillars have worked together to solve the COVID crisis, it acts as kind of a model for biotech innovation in the future? And do you think we'll see this kind of speed become more normalized, and this kind of cooperation become more normalized?
Well, that's a really interesting question. Let me sort of give you a couple of thoughts on that. First, I think what happened with the COVID vaccine is remarkable, and I agree with you in terms of the partnership. But certainly, in every other aspect of the COVID crisis, I would say it's been a disaster. I mean, you look at and it's tragic, really, right? You look at the diagnostics, you look at the mask issue. I mean, I would love it if at many, many levels. I mean, so I mean, I don't want to give myself or anybody else any credit about Moderna, but really that's the exception, right? Basically, when we started Moderna, all these people in finance actually would, and the newspapers and everybody said Moderna is going to do terrible. If you're doing vaccines, it's a terrible business. So, so that happened repeatedly. There were these reporters at Stat and other places that would just keep saying Moderna, even when the valuation was couple $1 billion. Now it's 70 billion, but they'd say they're never going to go beyond that. They were just huge amount of criticism. Also, now to go to masks and diagnostics, as far as I can see, there have been no incentives, no good companies. So you have one, maybe two, doing vaccines. Well, in diagnostics, we've had a total disaster as far as I can see, still is. If you go to anybody, I don't know if any of you have ever gotten tested. I think my son, his girlfriend at the time went to get a test. I think she got it back 5 months later. I can think of example after example of how bad the tests have been, and they don't work. There's lots of false positives, lots of false negatives. I mean, it's been terrible. Masks, I think, are also really important beyond, first of all, I mean, you had the CDC and the World Health Organization for months recommending to not use masks, because they'd say, well, it'd give people a false sense of confidence or other issues. Anyhow, and there have been, as far as I can see, there's, very tiny mask companies, but not really substantial ones. And it would be wonderful, I think, if the government or companies or investors felt like, boy, it'd be great to have a great diagnostic company or great to have a good mask company. But the fact is, again, if people created those companies, well, first of all, I think they'd have a hard time funding it. People also in the venture community generally don't like diagnostics. And a lot of times they don't like medical devices or masks. So I think it's really, really been difficult. And so you probably have to pick you have to probably pick the horses you ride on. So I feel that the vaccine thing, that I do think has been remarkably successful from the standpoint of getting the vaccine out fast. Now, even after that, I don't think the governments have done a very good job of getting it to the public. I mean, I think that was sort of a mess. I still think from what I see, it could be done a lot better. So I think that to me, the COVID vaccine Operation Warp Speed is, sort of, in my opinion, a stunning technical success, but it sort of stands alone.
Professor Langer, would you mind telling us a little bit more about the mRNA technology? Because I remember talking to a Princeton professor, Ning Yan, who is a very famous structural biologist, and I was talking to her, and even she was like, I really have no idea what the long-term effect or what this technology really is. And one of the kind of anti-vaccine narrative that is out there in the public these days is that this thing has been, invented in such a short time span. So who knows what's going to happen in 10 years out or whatever years out, and that kind of I guess eroded a lot of the public trust in the technology. So would you mind telling us a little bit how much we do know about this technology?
Well, I think you know a lot. I mean, first of all, like I say, this wasn't the first vaccine that Moderna put in the clinic. We've probably done over 1,000 patients on other vaccines, some who've had the messenger RNA for many years. So you have that database, number one. Second thing that number two, is just the science. So and Melissa Moore, who's at Moderna, I mean, she's one of the top people in the world in this, and this is what she told the FDA, which is exactly right, is that if you give messenger RNA, it doesn't last long. I mean, it's not DNA. It can't get integrated into the genome or anything. It's basically does what it does. It gets destroyed. It's going to be gone probably a week later. So to me, same thing with the nanoparticles. They're all going to be gone, very, very soon. So I think whether you look at it from a scientific standpoint or from the clinical data standpoint, it would be pretty surprising. I mean, I can't think of a reason. And like I say, a lot smarter people than I on the RNA area, like Melissa and also the people at the FDA, have looked at this, Tony Fauci, that I think there's every reason to think it should be safe. And I mean, and at least as safe as anything that people have ever done before. Now there will be more and more data coming over time, but when you really look at, just compare this to the flu vaccine, I mean, there have been, no more bad reactions to this than to the flu vaccine. And now many, many millions of people have been vaccinated. In terms of the long term, like I say, I just can't, I'm not trying to say that you couldn't have some person have bad effect. But the question is, would it be from this? In other words, if you look out of 20 million people, if you take any 20 million people, you'll find some that might have a reaction to this or that. But, but maybe it's not because of this or that, right? Maybe they breathe something in bad or something else happened. So I think statistically, there hasn't been anything that I've seen that's shown that any of these messenger RNA vaccines have caused any harm. So, and like I say, scientifically, I wouldn't expect it to.
So I think about time to start wrapping it up at this point. I wanted to bring it back to a question that maybe a lot of the viewers of this podcast can relate to. So you have often said that research is about asking the right questions rather than knowing the right answer. So what does this mean to ask the right questions, and how do you teach students to do this? And if a student comes to you today with a task that you think is impossible or infeasible, what do you would generally advise them to do?
So, so basically, if I think it's impossible, well, first, I don't ever tell any, the question is it really impossible? And the way I think about it is, first, is it a good idea? And secondly, what advice should I give them? And I guess what I try to say is that, have you thought this through? I think this is, I don't want to be over negative. I'd want to say, well, there's, some really good aspects. Have you thought about how long this might take? Do you have a plan about how you get there? What will you do if this happens? Things like that. You ask different questions.
Professor Lang, one question I'm pretty curious about is, do you ever think about philosophical questions as you venture through science? Because Alan Turing was famous for publishing philosophy journals and debating with Wittgenstein. And it seems that there's a lot of overlap between the scientifically and metaphysically unanswerable questions. And I was just wondering, what kind of things do you think about every day as you go on a treadmill or take a shower or have some kind of other free time to do other readings?
Well, I mean, it varies all over the place. I try to, I don't necessarily try when I'm, running or when I'm showering, but, sometimes thoughts creep in, and then they creep in for different reasons because, it could be something I've read. It could be just putting two different things together, from, talking to somebody to watching a TV show to listening to music. It just, the one thing that I've had positive going for me is I've learned, by being an undergraduate and PhD in chemical engineering, I learned something about that. And then being a postdoc in a surgery lab in a hospital, I learned something about that. So a lot of what I've ended up doing is sort of taking those two, and then sometimes something else triggers some idea, like the microchip for drug delivery or new materials that could change shape, all kinds of things. But it's maybe putting different things together in different ways.
Do you encourage, so if students come to you with I think maybe what is a common dilemma, which is going into academia or going into industry, how do you advise students? I think personally, I definitely am kind of still conflicted on that question and whether I would see myself more in academia or industry. And so I'm curious how you personally have thought about it and how you advise students when they presumably come to you and ask about it.
Well, I think both are great. I think it's really following your heart. I think that if you're in academia, you have enormous freedom. You're your own boss. I think that's great, but you're going to have to probably spend a lot of time raising money. And if you go into companies, I think that's also terrific. That you can really get a lot of fulfillment out of creating products that can hopefully change people's lives. You won't have to worry about the funding, but the company, you're going to have bosses in a different way, that have control over you. That not to say that academics is perfect, but but I think you have a lot more. Academics has more freedom, I think, in certain ways.
And then just to follow up more philosophically, it seems like the question that has guided you deeply throughout your life is what can I do that'll have an impact on people and make a difference in the world? Do you think that that's a question that's important? Like if a student were to ask you, what is the question that should guide my life? Is that a question that you think that you would you respond by giving them this question, or do you think that people think about it? Some people might think of, I want to really discover, and some people might say, I really want to help people. Is that a dichotomy that exists, or do you think that everybody should kind of think about how they want to make an impact on the world and how they want to make people's lives better? Like, is that an important guiding question for everybody to think about?
It's an important question that I think about, and I think sometimes it gives people a lot of fulfillment to do that. So I always think about impact, but really happiness is the most important thing. Different people, happiness and health, I think, are the most important things, and being a good human being, so I don't know that there's just one way to look at it.
Professor Langer, I know we're almost at 1 p.m. So just to wrap up here, 2 quick questions. One is, are you optimistic or pessimistic about where we're headed as a society? Because we have seen tremendous achievements in science over the past year, but also polarization, a lot of distrust in science. The second question is, because the name of our show is Policy Punchline, what would your punchline be for this interview? We've talked so much about your career and research. So,
well, I'm concerned about what's been happening, as you alluded to, but I'm always hopeful that the good of people will win out over the bad. And so I'm hopeful that things will get better, and that people become more aware because of what we've seen. But we certainly have work to do. And in terms of, to me, I always think, well, what would my punchline be? When I give like a commencement address or something, I always, I'm a dreamer. So I tell people, to dream big dreams that can change the world. Recognize that if you do that, you'll probably get a lot of people telling you that you're wrong or it won't work. And so I tell them not to give up.
Professor Langer, thank you so much for joining us today. It's just been such a wonderful interview. I'm sure Arjun and Michael and I, we've all just learned so much. So thank you so much for taking the time.
Well, you guys did a great job. I wish you the best, and thank you. Terrific question.
Thank you.
Thank you.
Thank you so much. Thank you.
Thank you. Bye-bye. Thank you.
Well, this concludes this episode of Policy Punchline. Please follow us on policypunchline.com and YouTube. You may watch this on our YouTube channel and listen in on Spotify, SoundCloud, any of your preferred podcasting platform. This is part of our continued coverage of aspiring intellectuals, where we have some of the more fundamental scientific questions in humanities, in philosophy, and science. So thank you so much for listening today. You've been listening to Policy Punchline, a podcast generously supported by the Julius Rabinowitz Center for Public Policy and Finance at Princeton University. We would also like to encourage you to follow other podcasts produced by Princeton University, such as Politics and Polls by the Woodrow Wilson School of Public and International Affairs. Policy Punchline is intended to be informational only and does not reflect nor represent the views of Princeton University or the Julius Rabinowitz Center for Public Policy and Finance. For more information on subscription, donation, volunteering, or contact, please visit policypunchline.com. Thank you again for listening.